you have the rest of the time to do your revision for your examination. And so I will encourage you that if you haven't started doing serious studies towards your examination, it's still not late. After today, you should be serious and make use of all the materials that are available to you, the lecture videos, the presentations, the book chapters, the self-assessment questions. They are all there for you, so you make good use of them. Because from today, you have more than 2 weeks, because I know your exam is starting 15th March. So that if even within this last week you have other lectures going on, or even going into next week, at least you have some 2 weeks for you to do that revision. All right, so we started with biotransformation in our last lecture, and we did particularly pharmacokinetics. Today we'll continue to look at pharmacodynamics, and that is where we'll close the curtain on MLB 203, Biochemistry 2. So previously we did look at the differences between pharmacokinetics and pharmacodynamics, and we simply said that pharmacokinetics deals with what the body does to a substance that is taken into the body, and pharmacodynamics deals with what the substance does to the body. And so today what we'll be looking at is pharmacodynamics, which covers dose response relationships. We'll take a look at some dose response relationship. Drug receptor interaction is just similar to what we did with the hormones, because when we were doing hormones, we did talk of cell communication. We talked about receptors on the surface of the cell. Those are transmembrane receptors, and then internal receptors, which are in the cytoplasm of the cell. And so it's just a similar type of interaction that those endogenous substances, the hormones, how they interact with the receptors is the same way that you have drugs also interacting with these receptors. Indeed, I mean, drugs, if you have, for instance, analogs of hormones, it means that those synthetic substances, when they are taken into the body, they will be acting the same way as the hormones that are synthesized in the body. So it's a similar kind of interaction that the endogenous substances have with the receptors that the xenobiotics or the exogenous substances will be having. Then there is also some pharmacodynamics also deals with some therapeutic and toxic effects. We talked about this somehow in the last lecture. But as we go on, of course, we'll be mentioning things about therapeutic and toxic effects, so that will be okay. But mostly today, what we'll focus on is mechanism of action of some selected substances. I mean, to make it more interesting, so that we'll deal with some specific mechanisms of action of substances, which will give you a very broad idea of how these substances act in the body to have their effects, whether it's going to be a toxic or therapeutic effects, how that occurs. So mainly, this is what will be based on mechanism of action. So we'll look a bit at dose-response relationship. But these two others, actually, we have looked at them even when we were dealing with other topics. As I said, for the drug-receptor interaction, we saw receptors and how they act, and therapeutic toxic effects. We did look at, when we were looking at this kinetic curves, we did talk about, say, safety corridor, where we look at maximum concentration of a substance and its effects, the minimum concentration, and the interval between them, which was the safety corridor within which a substance will be having its therapeutic effects. If the concentration goes above that threshold, then it begins to have a toxic effect. If the concentration is below the minimum, then it doesn't have the intended therapeutic effect that it should have. We did look at those two. So we'll look at the first, which is dose-response relationship, and then we come to focus on mechanism of action. All right. So this is to compare, this dose-response relationship is to compare, say, three substances: substance A, B, and C. And we see on the horizontal axis is dose. You can say concentration of those substances. And we have on the vertical axis lethality. And that is how the substance results into death. You can even replace this lethality with, say, therapeutic effects. If you just write the effect, it can be therapeutic effect, it can be toxic effect, but maybe since we are talking much more of drug substances, you may want to put the therapeutic effect. Anyway, it's medical science in a way, so it's understood whether you put effect percent or you put therapeutic effects or you put toxic effects. It's understood. All right. Now, if you pay close attention, you realize that those ranges have been divided into 5 different regions. We have region 1, region 2, region 3, into 4 regions, and region 4. So it's divided into 4 different regions. And what you notice is in the first region, region 1, if you are comparing the effect of substance A, B, and C, then clearly you see that substance A is having the highest effect, followed by substance B, and substance C is having the lowest of effect in that first region. Now, if you move to the second region, you realize that that trend, which was in the first region, is not the same in the second region. What you notice, the effect of substance B has now become the highest, followed by substance A. Substance C continues to be that substance that is having the lowest of effects. You move to the third region, you notice again there is some difference. Now substance A is having the lowest of effect. Substance B and C somehow look like they're having the same effect. They sort of intersect along the line. But even that within the same region, somehow you could see that at a point you could have the differences between substance B and C, but at a certain point, substance B and C are having the same effect because they intersect there. Now, why are we doing all this? If you look at by the time you get to the fourth region, you realize substance A is having the least effect. Substance B is having, the intermediate effect, whilst substance C is having the highest effect. So the question again, why are we, looking at these different dose ranges? If you are doing, for instance, some maybe you are developing a drug substance, for instance, you are comparing different substances that have a possible effect that you want to take advantage of, exploit that for maybe some particular therapeutic effect. And imagine that you take the same, concentration of these substances, and you just test it on. Usually you do this, and those of you who like this microbiological test, you usually will have some culture. You introduce this substance into the culture, and then you begin to have some effects. You measure those, count and things like that. Now imagine you are having a substance which you are introducing not only into culture, but maybe you are doing some animal tests, and you take concentrations of the substances within the first range, and that is all. Like you just test this, and you say, oh, substance A is having more effect than substance B, and substance C is having the least effect, and so you choose substance A as the substance that is having, say, the best of effect there. What this means is that what this whole relationship is telling you is that may not be the whole picture. In a certain region, you can be having such trend, but if you move into a different dose region, you'll be having a different trend. So this is a way of actually studying the full spectrum of the activity of substances before coming to a conclusion of which of the substances is having the optimum effect that you can exploit in drug development. So you can see clearly that if you actually look at the full spectrum of how the substance is acting, then you will be able to have that a complete comparison of different substances than just choosing some concentration and testing, and then you just quickly decide that, oh, this is better than this and this is the best. So I think this one is very much straightforward, so you can understand what it is that you have to study a full spectrum in terms of comparison of substances to come to a conclusion on which of the substances is having the optimum effect. Now, still continuing with dose response relationships, there's another thing you do when you are particularly wanting to establish permissible limits of toxic substances. What you need to do is also to determine that, apart from you checking for the effect of the drug substance generally, like say in a general population, that you see that you have the dose response curve, which the main dose response curve is this curve number one, with which you see there is what is being referred to as a background effect. That is to say that below that level of concentration, or below that uh, uh, below that line, you have the concentration of the drug or the dose of the drug not having, any significant effect, so that it is only above that background effect that you begin to notice or have some significant effect, whether it's therapeutic, whether it's toxic effect. It is only above that background effect. It's just like the way you do like blank, so that the concentration of a substance between, a certain range to a certain range really does not have any effect at all. It's so minute that it's negligible. Say nothing happens, so that it is above a certain threshold. And you see there is an intersection between the curve and the background effect. That intersection is what is being referred to as a practical threshold. So that it is at that practical threshold when you have the dose or the concentration of the substance increasing from that practical threshold, you begin to have significant effect of the substance until it gets to a point that is constant. I mean, the 100% of the effect is there. And so even if you increase the concentration, the effect will just be the same. It's 100% already. But there's something else. You see that apart from curve 1, there is curve 2, there's curve 3, and there's curve 4. And what are these? So in a population, even though you have the main effect of the substance that you are seeing to be the curve 1, there is a possibility of, the substance, even at a very low concentration, having some non-threshold dependencies in some people. So some very few people, even at very, say super small concentrations, such persons have some significant effect. So that is possible. So that you have this, curve 2 is showing, in that there are some persons who are even more sensitive to that substance, such that even at such a very small concentration, they quickly, begin to react. In some persons, such, threshold effect is very low, and so you see, in such a case, you have a practical threshold for such super small concentrations. But in studying these things to determine the permissible limits of substances, particularly when you're dealing with working environments, for instance, or even residential areas that, sometimes you want to determine permissible limits of substances that can be in those places. Theoretically, the straight line, the curve number 3, is used for such estimation. So that that permissible limit is determined as the intersection between the straight line and the background effect. Now, lastly, you see there's a value here, LD50, LC50. So these are doses or concentrations that result in 50% of the effect. Usually, what this means is that, when some animal tests are being done. Usually they use a certain number of, laboratory animals like mice, rats, to do some analysis of these substances, and the dose at which 50% of the animals that are used for these tests die, that dose becomes the LD50. In other words, the concentration of the substance that will result in, let's say for instance, you take 10 mice and you test a substance on them, where a particular concentration of the substance on 10 mice, and 5 of the mice survive, whilst 5 of the mice die out of that concentration that is given to them. Then that concentration becomes the LD50, because it is at that concentration that 50% of that population, the mice population, died. Sometimes you see LC50. LD means dose, lethal dose. LC means lethal concentration. So they refer to the same thing, either LD50 or LC50. They refer to the same thing. All right. Now, we want to take a look at, there's this common thing that a lot of times when I sit on your project work panels, I notice some of you do some extractions, and you do some tests, particularly microbiological tests, and then you do some mixtures, different extracts, and a lot of you refer to the term synergistic effect, which really in most cases, what you mean by synergistic effect is not really what it is. So pay attention so that here, you have a very clear understanding of what synergism is, what synergistic effect actually is. Because I noticed that it's a common mistake that the students make all the time when they come for their project defense. Now, first of all, if you take substance A and B, you have substance A on the left in this diagram. So you have the effect of substance A, which is the vertical axis on the left, and you have the effect of substance B, or the vertical axis, on the right. And on the horizontal axis is the amount of substance. So that's going to be substance A or B, the two of them as a mixture. You see a curve number 1, where you see there is that gradual increase when you put these two substances together, substance A and substance B. And you notice that curve number 1 is corresponding to what we refer to as summation or additivity. So summation effect or additivity. What simply that means is that if you take substance A that is having a particular effect, particular therapeutic effect, and you take substance B, which is also having the same therapeutic effect, and you put them together, their additive effect, their summative effect, the effect of the two of them together is what you are referring to as summation effect or additivity effect. That is represented by the curve 1. Now, what is potentiation? So Potentiation, in this case, also referred to as synergism, is that if you take substance A, which is having a certain therapeutic effect, and you take substance B, which is not having that therapeutic effect. It's not having any therapeutic effect. Substance B is only increasing the potency of substance A. So you see, you cannot talk about synergistic effects of two substances. So what I mean is you can't say synergistic effect of substance A and B. No, you can say synergistic effect of substance B on substance A, meaning substance A is having a certain effect and substance B is coming to increase that effect of substance A, or vice versa. So you take substance B that is having a certain therapeutic effect. Substance A is not having that therapeutic effect. But when you introduce it into the presence of substance B, it increases the potency of substance B. And so you can say synergistic effect of substance A on substance B. So I hope you get a difference. You can't be saying synergistic effect of substance A and B. No, you can say synergistic effect of one substance on the other. Or whether it's a combination of maybe two substances are having a synergistic effect on a particular substance. You can talk about it that way. All right, so clearly you should now see the differences between summation additivity, which is and then potentiation, which is synergism. Now, antagonism is the opposite of potentiation or synergism. So that if one substance reduces the potency of another substance, then that substance is an antagonist. The substance which is reducing the potency of the other is an antagonist. So that one too, I think, should be clear. Antagonism being the opposite of potentiation or synergism. All right. Now, a bit more of revision. We did see this already, where you have a signaling cell that is releasing some ligand. When we were dealing with hormones, that ligand was a hormone, and then a target cell was having a receptor. That receptor can be a protein, it can be some glycoprotein, can be some glycolipid, so some biomolecule which is embedded in the membrane. We even saw that there could be receptors inside of the cell, so that some substances can penetrate through the cell membrane into the cell to interact with the internal receptors. But here we want to remind ourselves that receptors are selective. Receptors interact with particular ligands, so which means that specific drug substances are going to interact with specific receptors. And then we also have what we refer to as silent receptors and active receptors. So a silent receptor is a receptor that is not going to cause any response if a substance binds onto it. I mean, you can even take a rough example of, say, blood proteins, like, say, albumin, that binds to some substances. So in that case where we talk about bound substances, and even we then infer to say bioavailability, to say that the amount of the substance which is unbound, which is free flowing in a bloodstream, is what is active, and that will amount to how much of the drug substance is available to have its effect. Even if you take endogenous substances like when we were looking at the metabolism of bilirubin, and you saw at a point bilirubin bound to blood albumin, that protein. It didn't result into any effect. So, I mean, it's a similar thing of the sort that you have substances that can bind onto certain receptors, but that will result to no effect. Those are silent receptors. How about active receptors? So an active receptor are those receptors that when a drug substance binds to them, it will stimulate an effect. So those are active receptors. Okay. Now, the simplest theory, the simplest form of studying the effect of substances based on receptors was introduced by the English pharmacologist Joseph Clark, Professor Alfred Joseph Clark. He was a British and was in the Royal Army. I mean, he served in the First and Second World War and actually worked in the University of Cape Town in South Africa some time before going back to Britain. And he wrote a lot of books in pharmacology, pharmaceutical chemistry. And he was the first to bring about a theory that was the first attempt of quantifying receptor effects. What is being referred to as the Clark's occupancy theory, sometimes being referred to as the receptor theory. And in that, what it refers to is that the effect of the substance is proportional to the area of receptors that are occupied by molecules of the substance. So what simply it means is that if you have a substance that is bound to, say, 5 receptors, it will have a certain effect. If you have that substance bound to 10 receptors, the effect that it's having will increase. If you have it binding to 15 receptors, the effect will increase the more. So this is also to say that you have a certain dose or concentration of a substance, and as you increase the dose or the concentration, the effect of the substance is going to increase. And why is the effect of the substance increasing? The effect of the substance is increasing because more of the molecules of the substance are binding to more receptors. And again, if you take 2 substances, for instance, because another thing is that it says the maximum effect of a substance is realized when a minimum number of its molecules are able to bind to and disable or stimulate vital target cells. What it means is that if you have 2 substances, so let's say you have substance A and substance B. Now, let's take for instance that you take 0.1 mole per liter of substance A, and it produces a certain effect. And in order for substance B to produce the same effect, you have to use, say, 0.3 mole per liter of substance B in order to get that same effect that when you take 0.1 mole per liter of substance 1 is giving you. Clearly, what it means is that substance A is more potent than substance B because you use a less concentration and you got an effect that for substance B, you have to take, say, 3 times of that concentration before it will produce the same effect for you. And so the lesser the amount of the substance that is stimulating a particular cell to have a certain effect, the more potent that substance is. All right. Now, when you take any drug substance, that drug substance will fall into one of these mechanisms of action. It is either you have that drug substance that can act directly on an invader. Let's say an invader is a bacterial cell or maybe some virus. So it's either you have the pharmaceutical agent or the drug acting directly on the invader. Now, how does it, act? By what mechanism on the invader? You can do that in two ways. One is to distract the membrane structure of, say, the bacterial cell. So that means that it can destroy the membrane structure of the bacterial cell. In other words, that is going to result into the bacterial cell death. That's what you refer to as a bactericidal effect. Because once the membrane integrity is disrupted, what it means is the cell is going to break, and you have components of the cell just spreading, and that's it. The cell will be no more. Then you can also have that drug substance not destroying the cell, but rather targeting some specific metabolic enzymes, so that it will disrupt a metabolic process of the bacterial cell. That's what you refer to as a bacteriostatic effect. So it can inhibit the growth of the bacteria or inhibit, say, reproduction of the bacteria. So it will do that by, say, maybe inhibition of some enzymes that are involved in some metabolic processes of the bacteria. So those two things can be the way a pharmaceutical agent will be acting directly on an invader. Now, a drug can be acting also directly on the host cell. And these two can be by different mechanisms. One, the drug substance we have seen already, like, when we're looking at cell transduction, we saw this kind of mechanism where you had a receptor, a transmembrane receptor that got bound onto a ligand. And there was a second messenger that transferred that message. That whole process we are referring to as signal transduction, all the way until you have the message going into the nucleus to have some transcription and translation of a particular gene, which will produce some protein, which will then become some particular maybe enzymes or neurotransmitters, hormones, resulting in an ultimate biochemical response. We saw that process. And so here, you can have this drug substance coming to bind onto that receptor. So that when it binds onto that receptor, it's either it stimulates the receptor just like how the maybe endogenous substance that binds onto that receptor stimulates it, so that there will be a biochemical response. So a drug substance can be an activator of that receptor to stimulate a particular signaling to occur. Or it rather comes to block the receptor. It binds onto the receptor and prevents the ligand which should bind onto that receptor to produce an intended signaling. Or second, what can happen is you can have a drug substance that is binding onto a transport protein. So in the cell membrane, you also have transport proteins that allow for the transport of substances, mostly precursors for different metabolic processes to occur. And you have a drug substance that comes to bind onto that transport protein and causes it to close. That is, causes it to prevent the transport of such precursors for the metabolic processes, which could be some biosynthesis that should be occurring inside the cell. You can also have the drug substance targeting metabolic enzymes. So the enzymes that we did look at some enzymes, so that are involved in processes of usually biosynthesis of molecules like proteins, carbohydrates, lipids. You have the drug substance coming to inhibit those enzymes, and thus will be inhibiting those metabolic reactions. We also have what we refer to as ion channels, which are also in the cell membrane. And these act, some of them act as what we refer to as voltage-gated ion channels. So these act on difference in potential across the cell membrane when there is movement of ions across the cell membrane, and it creates a potential across the cell membrane. That is when you have a positive charge at one side of, say, the cell membrane more than the other, and vice versa. You have negative charges at one side of the cell membrane or the other. So this can happen when you are having, say, movement of, say, hydrogen ions, from the cell to the outside of, to the extracellular space. Then if there is no compensation for that, what can result is that you have more positive charges outside than inside. So those channels work by the changes in the potential across the cell membrane. That's why we refer to them as voltage-gated ion channels. You do find them in the cardiac system, acting in this process of contraction, relaxation of muscles. It's not only the voltage-gated ion channels in a way. We also have what we refer to as the ligand-gated ion channels. So that these ones, they will be stimulated by biochemical substance that comes to bind onto them for them to open or close. So you have those ligand-gated ion channels. You have also voltage-gated ion channels. Now, this one too, you can have a pharmaceutical agent that is coming to either activate or block the activity of those gated channels, preventing the movement of ions, like, say, calcium ions, potassium ions, hydrogen ions. And that will result into the potential across the cell membrane, which will then result into contraction, relaxation of muscles. All right, so you see these different ways by which drug substances can be acting, either on the an invader or on the whole cell. Now, what to do is we'll take some specific examples of drugs and look at how, they act. So we'll take some mechanisms of action. Uh, After we've looked at that, broad overview of drugs, how they could be acting, I want us to look at some specific examples. Now, the first we'll look at are some antibiotics. But in order for us to have an understanding of, how these antibiotics do act, we first take a look at the process of the biosynthesis of tetrahydrofolate, what you refer to as folic acid. So we do synthesize folic acid in the body, and bacteria also synthesizes folic acid. Folic acid is needed for cell growth, cell proliferation. I mean, that is the reason why folic acid sometimes is given as supplements to, say, pregnant women, so that there will be no deficiency. Is very much required for that embryonic growth, where you have such high level of cell growth, cell division, cell specialization, proliferation. So you do need folic acid, particularly for the growth of the central nervous system, the brain. The deficiency of it can result into deficiencies in the growth of neural tubes. I mean, simply in the growth of the development of the nervous system, particularly the brain, which, if that deficiency is not checked, can result into some mental retardation. That's why it's very much needed, and sometimes being um, prescribed for pregnant women. So yes, the same way as you have this very essential tetrahydrofolic acid being important in humans, bacterial cells also do use this uh, hydrofolic acid, tetrahydrofolic acid, for their cell growth, cell proliferation. And now, the process of synthesis of this tetrahydrofolic acid is from two components. From the beginning, you see you have a dihydropterin pyrophosphate. That is the ring, the two ring structure, the red molecule. And then a blue molecule, 4-aminobenzoic acid, these two molecules. In the first reaction, what you see is that these two molecules conjugate to form dihydroteric acid. So it's very simple here. You can see red molecule, blue molecule, they conjugate and they form dihydroteric acid. Then in the second reaction, what you see is that there has been a conjugation of a green molecule. That green molecule is glutamate, an amino acid. And then from there on, what you notice, the next thing you see is a reduction reaction. And so if you pay attention, you realize that on the second ring, that nitrogen is having a double bond there. And that double bond is removed in the tetrahydrofolic acid. And there's a hydrogen on that nitrogen. Previously, there was no hydrogen there. So simply what it means is that two hydrogens have been added into this molecule. One on that nitrogen, the second will be on the carbon that was having a double bond with the nitrogen. So you have a reduction reaction there. So simple reactions to depict to just simplify the synthesis of tetrahydrofolic acid. Now, let's pay attention. There are some very small notes here. There's a small note here. There's a cross on this first arrow, and there's a small note there that says sulfonamides. So sulfonamides are a group of antibiotics, which means those antibiotics can inhibit the enzyme that catalyzes this reaction. The same way, on the last arrow here too, you see trimethoprim. There's also a cross there. Also can cause inhibition of this reaction. Now let's zoom in. Let's zoom in on those reactions with those small notes there. So the first reaction, sulfonamides, also sometimes being referred to as sulfanyl amides, act by inhibiting the enzyme dihydroterate synthetase. This enzyme, which is responsible for catalyzing that first reaction, the conjugation of this red molecule, dihydroterine pyrophosphate, and this blue molecule, 4-aminobenzoic acid, to give you dihydroteric acid. So if sulfonamides inhibit this enzyme and this first reaction does not occur, what it means is that the synthesis of the tetrahydrofolic acid is going to be inhibited so that you don't have enough tetrahydrofolic acid being produced in the bacteria. That is going to slow down, is going to inhibit bacterial growth. So that is going to be a bacteriostatic effect. In this case, if you look, you see the structure of the sulfanylamide and para-aminobenzoic acid, which is the same as 4-aminobenzoic acid. So if you see 4-aminobenzoic acid and para-aminobenzoic acid, is the same name. Para only means the amine group and the carboxylic group are opposite. They are opposite each other. And if you say 4-aminobenzoic acid, the carbon on which the carboxylic acid group is carbon number 1. And if you count, the next carbon, carbon 2, clockwise, the next is carbon 3, and the next carbon 4, that is on which you have the amine group. That's why you see it's also being referred to as 4-aminobenzoic acid. Now you see the structures, they are similar. And I think we did look at mechanisms of action of enzymes, and we referred to enzymes acting as, we referred to substances acting as inhibitors to enzymes. We look at one of these inhibition processes as competitive inhibition. And so here you can see with this structure of the antibiotic, it looks much like the para-aminobenzoic acid, and so can bind into the active center of that enzyme, dihydroteric synthetase. So it's like so it has a structural analog of para-aminobenzoic acid, and it will prevent that reaction from occurring. All right, now that third reaction, also we zoom in and we see that from the dihydrofolic acid, this is where the reduction occurred, where the double bond was removed. Hydrogen atoms were added into the molecule. And you see another antibiotic, trimethoprim, also inhibiting the enzyme dihydrofolate reductase, which is responsible for catalyzing that reaction. And so the synthesis of tetrahydrofolic acid is also inhibited there. So that too is going to be a bacteriostatic antibiotic there. And if you look at the structure, not so similar to the structure of the substrates, dihydrofolic acid. So this is where you do talk about broad spectrum antibiotics. I mean, the more closer the structure of the drug substance is to the substrate of an enzyme that is blocking, the more specific that drug substance is going to be. So we can look at another one, which is that same reaction. You have another drug, methotrexate, which is actually used in chemotherapy to inhibit the growth of cancer cells. And it's also inhibiting that enzyme, dihydrofolate reductase. And if you look at the structure, you see that the structure looks very, very much like the substrate dihydrofolic acid, which means methotrexate is going to be a very, very specific antibiotic than the one we just saw, which was broad spectrum. This is going to be very much more specific because if you look at the structure, it looks very, very much like that of the substrate. And you ask a question, so if you are having these drugs, somebody says the network is not stable. Is it just now? I hope that from the beginning it was okay, and it's just along the line that it became a bit rough. All right, so I was saying that you could then be thinking, so if you are having these antibiotics sort of inhibiting the synthesis of tetrahydrofolic acid, and you have the host cell, say the human cell, is also using this same process for the synthesis of the hydrofolic acid. So if these substances are introduced into the body, won't it affect, the synthesis of folic acid of the host as well? Yes, so that is a side effect that you can have sometimes. So for instance, methotrexate, which is used for treatment of cancer, because of that effect that it can also have on healthy cells, normal cells, not only on the cancer cells. That's why you have it resulting into some hair loss, some scaly skin. So you do have, yes, such side effects as well. Even though the dosage is such that it will inhibit particularly those cancer cells from growth and proliferation, but you will still have some those there that will affect the normal cells as well. All right, now we take another group of antibiotics in the penicillium family, and there too, before we move to that, we need to understand the structure of the gram-positive bacterial cell wall. So if this gram-positive bacteria, and if you zoom in on the plasma membrane and the cell wall, you realize, yes, there's a plasma membrane, which is a phospholipid bilayer. And then you have the cell wall, the very dark, thick part of the cell is what is being shown by what we refer to as the peptidoglycan layer. The peptidoglycan layers are These are some polysaccharides that are, well, or maybe you can even refer to it as oligosaccharides with some peptide linkages. So that's why we say it's a peptidoglycan, carbohydrates and peptides at the same time. So we have these violet balls referring to N-acetylglucosamine and the blue N-acetylmuramic acid. Then you have these dash lines, what you see in between these oligosaccharides, referring to some peptide chain. It said some 4 amino acid, peptide. That's a tetrapeptide chain cross-linked to hold these oligosaccharides together. So that is the general structure of the peptidoglycan. So in the peptidoglycan structure, you have these modified monosaccharides, N-acetylglucosamine, N-acetylmuramic acid, and you see them alternating. Then you see that there is a tetrapeptide, 2 tetrapeptides that are binding this N-acetylmuramic acid, residues. So you have such cross linkages between these. So it holds this whole structure together that way. All right. Now let's move on to see the process of the synthesis of this peptidoglycan. So starting from the top, you have the light green or teal, and then say the deeper green. They are referring to this N-acetylmuramic acid and N-acetylglucosamine. So the deep green there is N-acetylmuramic acid. The light green is the N-acetylglucosamine, and so it alternates. These oligosaccharides, they alternate. And you also see that on N-acetylmuramic acid, you are having some short chain peptides. You have these short chain peptides there. Now there is a protein here that we are referring to as the penicillin binding protein. The reason why we're referring to it as penicillin binding protein is because that is the enzyme that the antibiotic penicillin targets. But if you want to refer to it in terms of its function, what it does is a glycopeptide transpeptidase, because it's responsible for the cross linkages between the peptides. And so you see, first thing, that penicillin binding protein, you notice is having in the active center serine. It's a serine residue. Now you see the protein comes to bind onto one of these peptides that is binding to that serine, amino acid serine in the active center, and into the short pocket. There are 2 pockets, one is short and the other is long. Now after it binds, you see there is a second chain of this glycopeptide. You have this chain with another short chain peptide that comes to bind into the longer pocket. And the next thing you see that there is a cross linkage that is formed between this lysine residue, and the red is alanine. The keys are here, so you can look at them. Red means alanine, this blue means glutamic acid or glutamate, and the green means lysine. So you can easily relate and see that that cross-linkage is between lysine and alanine. After that, you see the enzyme dissociates. So the cross-linkage is formed. So this is just to show how the cross-linkage is formed and the fact that the enzyme which is responsible for the formation of these cross-linkages is the glycopeptide transpeptidase. All right, now we have penicillin, which can inhibit this protein, this enzyme, the glycopeptide transpeptidase. And because it is this protein that penicillin is targeting, we refer to it as penicillin binding protein. So all that process that we saw from the beginning is what is, being depicted here from the top coming down. But here's the thing, we see these structures there that are like 6 or 9. That is, to show penicillin. So those are like penicillin molecules there. And what do you see? You see that in the presence of penicillin, or when you introduce penicillin, the penicillin molecule reacts with that reactive serine in the active center and opens up and goes to block the active center of the enzyme. And once it blocks the active center of the enzyme, it means that there will not be the formation of these glycopeptide, the peptidoglycan, which is the cell wall of the bacteria. So that is how the cell wall of the bacteria can be disrupted. The synthesis of it can be disrupted, so that penicillin also becomes a bacteriostatic antibiotic. Now, if you look at the structure of penicillin, so this is penicillin structure, which is here. As you can see, it's a closed ring. The rings are closed, just like it's being depicted here, like 9 or 6. The rings are closed. And if you look at those peptides that are joining to give you the cross linkage, the structures are similar, but you see that because these are peptides, they are open. There's no closed ring there. Penicillin is closed, but when it comes into contact with the enzyme, it reacts with that reactive serine in the active center. Then the ring opens, and then it binds in there. Sometimes being referred to as some suicide inhibition, because it's like it's inactive. But the moment it comes in contact with the enzyme, then it opens up and binds to the enzyme. All right, now you see this ring. Let's pay attention to this ring, which we are seeing here in penicillin, because we'll come back to it. So the process we've looked at so far, you can simplify that if you have this as the glycopeptide transpeptidase, and the active center is having a serine residue. The serine is having a side chain of CH2OH. If you go back and look at the structure of serine, the amino acid serine, and you see penicillin comes to bind on that active center, serine, and blocks the enzyme from carrying out its function of the formation of the cross-linkage in the formation of this peptidoglycan, which is the cell wall of bacteria. So this complex, the penicillyl enzyme complex, renders the enzyme inactive. Right. Now let's go to the ring. I mean, you see, I think you've been hearing something like the beta-lactam antibiotics, which is I think one of these common antibiotics which are produced in Ghana by some of these pharmaceutical companies. I think if you go onto the website of, say, is it Entrance Pharmaceuticals or Tobinco and some of these pharmaceutical companies there in Ghana, you'll see they talk about beta-lactam antibiotics in those places, in those websites, because they do produce those. That's the ring, the active ring, which is in penicillin. And you have different types of them. So you see looking similar like what we saw with penicillin, similar rings. The first here we refer to as the PNAM ring. Following it is the PNAM ring. If you look at it, they look similar. The only difference is there's a double bond here for the PNAM ring that you will not find in PNAM. And you also see carbapenem. I mean, so in place of this, the sulfur you were seeing there is just a carbon. So it's a carbapenem. And you have CFAM. So now it's no longer, it's not a 5-sided ring. It's now a 6-sided ring. CFAM, CFAM. So CFAM is having a double bond there, which is not in CFAM. So you have these different beta-lactam rings. I mean, the common drugs you find in Ghana, amoxicillin. So if you look at the structure of amoxicillin, it's having that ring there. And we have this combination of amoxicillin and clavulanic acid. You see, clavulanic acid is also having similar ring. That ring is not here. In amoxicillin, the ring there is the first one, which is the PNAM ring. But in clavulanic acid, that ring you don't find here there because there's an oxygen there. That's an oxypenam. So actually, you have these rings are actually not exhaustive. There are more of them. That if you go online, you'll find the different types of the beta-lactam rings. But clavulanic acid is combined with amoxicillin to give you the common drug we have on the Ghanaian market to refer to as amoxiclav. And that is to deal with the bacterial resistance to amoxicillin, because clavulanic acid inhibits an enzyme that bacteria produces. I think is it clavulanase or so, that it uses to for its resistance against amoxicillin. So when you combine this clavulanic acid together with amoxicillin, it then deals with that bacterial resistance against amoxicillin. So I have that combination. All right. Now, I think we just pay attention to the first two reactions, which is, but we have seen actually this reaction from diacylglycerol to maybe not arachidonic acid, but we saw something with diacylglycerol phospholipid. If you remember, when we were dealing with the hormones and we were talking about the second messengers, we did see one of the mechanisms in which you have some, the second messenger was inositol, triphosphates, breaking down to give you diacylglycerol and all of that. You saw that. But here, what you are seeing is that We have prostaglandins, some endogenous pain relievers. I mean, they are responsible for pain recognition. And the synthesis of them is from the lipid arachidonic acid. It's a 20-carbon fatty acid. And the arachidonic acid is produced from diacylglycerol phospholipids by some phospholipase. We did see this, phospholipase C, phospholipase A2. We saw some of these enzymes. Now you see this reaction from arachidonic acid to prostaglandin. You see that you have some enzymes there, cyclooxygenase 1, cyclooxygenase 2, what is being abbreviated COX-1, COX-2. And I think some of you maybe have been working in some pharmaceutical, maybe you did some attachments at some pharmaceutical company, or maybe even you went doing attachment in some hospital or so. You did see COX-1, COX-2 drugs, things like that. I mean, what it means is those painkillers which you see as COX-1, COX-2, or even if you buy any medicine and you are reading to like those analgesics, some of them will refer to, say, maybe inhibition of cyclooxygenase. And it's because those enzymes are responsible for the synthesis of prostaglandin from arachidonic acid. And it's these prostaglandins, they result into, say, maybe from prostaglandins, you have some of them being thromboxanes, which are responsible for some blood clotting. So that is when there is some inflammation. So that if you have that prostaglandin synthesis being inhibited, then of course this process of the production of thromboxanes is also going to be inhibited. Prostacyclins will do the opposite of thromboxanes. They will rather be acting as some vasodilators. So yes, you have this process that you have the cyclooxygenases acting. Now, a drug like brufen, common drug like brufen, also acts as an inhibitor to these cyclooxygenases, so that it prevents the production of these prostaglandins, which means less prostaglandins being produced results in less pain recognition, results into pain relief. Now, we do have aspirin also doing similar thing, where it also inhibits cyclooxygenase 1, which means in the end is going to inhibit the synthesis of prostaglandins, thromboxanes, and so will also result into pain relief. Now, we do see also maybe this is more or less the physical, not so much of chemical per se, because so you do have let's say some heavy metal poisoning, and these heavy metals, what happens is they bind to proteins and inhibit the activity of these proteins in the body. So what you see here is that you have a protein that a heavy metal is bound onto its soft. You have in the proteins, you have some the amino acid with sulfur in it is what? Cysteine. And where you have cysteine, you can easily have this heavy metal binding. So where there is some sulfur hydride groups, or maybe where you have some disulfide bridges, you then have these heavy metals coming in there. And those disulfide bridges, they actually participate in the functionality of proteins. You remember you do talk about the tertiary structure of proteins. which actually gives the protein its functional structure before, even if you have quaternary structure, that's going to be subunits of these functional proteins. And so if you have heavy metals that are binding onto these proteins and disrupting those disulfide bridges, obviously it's going to disrupt the functionality of these proteins. But then you then have this, what you refer to as diethyl antidote, that is having this also sulfur hydride groups, 2 of them. I mean, that can easily come in and bind to this heavy metal. So if you have the diethyl antidote just coming to bind onto the metal, it frees the protein. The protein is now free. And so you have this antidote that is forming a complex with a metal excreted out of the body. So it's a form of antagonism. It's an antagonism of an antidote. Another one is, let's take a look at I think this one actually, you did really do something on the mechanism of blood coagulation, even in the secondary school science. You did talk about the fact that you have platelets. So platelets in an inactive form, they get activated by a platelet activating factor, which is a lipid. And you now have these activated platelets. Don't bother yourself so much about all of this. These are like conversion of different, proteins, factors, which will then be activating. You know how we saw those cascade of reactions one after the other. One thing is activated and the other one activates. That's all what is being shown over here. We'll not pay so much of attention to all these details. But this happens in 2 phases: initiation phase, amplification phase. But I want you to pay attention to this place. You have factor XA. You can say factor 10A. So that's some particular protein that is responsible for the conversion of this protein, prothrombin, to thrombin. And after you have that active form, thrombin then goes into the process of conversion of fibrinogen to fibrin. And fibrin is going to form a mesh. So where there is some inflammation, you have fibrin forming a mesh which will trap red blood cells. That's going to result into formation of a blood clot. So for instance, if there is some wound on the skin, this is the process that occurs. You have platelets activated, a cascade of reactions occur, and you have this factor 10A converting prothrombin to thrombin, then thrombin converts fibrinogen to fibrin. Fibrin forms a mesh, traps the red blood cells, and forms a blood clot. That is going to stop the bleeding from the wound. Or you have thrombin even going into the conversion of a factor 13 to its active form, which then will also be acting on the fibrin in its stability. And so you then have that, trapping of the red blood cells, which will be resulting into blood clot. Now, having seen this mechanism of blood clotting, we have, say, anticoagulants, say heparin. So these are like 3 different kinds of heparins that their mechanism is similar. Is similar. So that what is happening is that heparin binds onto the protein antithrombin that is activating it. So heparin binds to antithrombin to activate it, and that activated antithrombin then binds to the factor 10A, that XA that we saw, which was responsible for the conversion of prothrombin to thrombin. Once heparin binds onto it, it deactivates it, it inhibits it, so that you don't have that conversion of prothrombin to thrombin, which means blood clotting will not occur. That is how the anticoagulant action is achieved by these medicines, like the heparin. All right, now we can take another one, antagonism by ethanol, for instance, where it's used also. So ethanol is used in the case of methanol poisoning or methanol intoxication, because the same enzyme, alcohol dehydrogenase, is responsible for the metabolism of methanol and ethanol, and it's responsible for the metabolism of alcohols. But when you have methanol binding onto that enzyme, alcohol dehydrogenase, it forms toxic metabolites. Formaldehyde, formic acid, those are toxic, that have adverse effects on the central nervous system. In fact, within a short time, it can result into blindness. And so if you introduce ethanol as an antagonist, what happens is ethanol goes to bind onto the alcohol dehydrogenase. And when it binds onto the alcohol dehydrogenase, it then prevents the binding of methanol, so that those toxic metabolites will not be produced. For ethanol, the enzyme breaks it down into less toxic metabolites, which is acetaldehyde, acetic acid, which will then go into the formation of acetyl-CoA, and you have seen acetyl-CoA in many biochemical processes like the glycolytic pathway, the TCA cycle. Mention them, metabolism of lipids, proteins. So that's less toxic. It will just go generate energy. So that's an alcohol being used for the treatment of intoxication of another alcohol. Methanol is toxic, but ethanol can easily be broken down by the alcohol dehydrogenase into less toxic metabolites. And so it can be used as an antagonist against methanol intoxication. I think maybe this should be the last one, beta blockers. So this is also much like what we saw with these receptors when we were looking at the hormones. Of course, adrenaline, noradrenaline were hormones that we looked at. And you remember this system of the second messenger, cyclic adenosine monophosphate. This is beta blocker. So we'll look at even though here is showing different receptors, alpha 1, alpha 2, and beta, but let's just look at the beta. So for the beta blockers, they will be acting on this receptor, this particular receptor, the beta receptor, which we have seen this reaction mechanism where after you have adrenaline or noradrenaline binding onto the receptor, it results into the conversion of ATP to cyclic adenosine monophosphate with the action of that enzyme, adenylyl cyclase, and cyclic adenosine monophosphate, several reactions, and you then have some muscle contraction, muscle relaxation. It also will facilitate the process of glycogenolysis, the breakdown of glycogen in the skeletal muscles in the liver to produce energy, to release oxygen into the bloodstream for more energy. And so it's a system which is active in the contraction and relaxation of cardiac muscles, smooth muscles. And adrenaline is being secreted from, say, these peripheral nerve cells. So as a neurotransmitter, which then will come to bind onto this beta receptor. So the beta receptors are embedded in the cardiac cell membranes, so that beta blockers, when they bind onto the beta receptor, they will then prevent the binding of adrenaline on the beta receptor. That is to treat condition of cardiac arrhythmia, particularly like high blood pressure, which means it will reduce the contraction relaxation of the heart muscles, to sort of reduce hypertensive effects. All right. So I think this is where I'll stop talking and open the floor. Anyone with any comments, any questions, any contribution, you can write in the chat, or you can raise your hand. I will enable your microphone. You unmute yourself and you speak. The floor is open.